Good evening. Welcome to today's session. Am I audible? Okay. So, in the last class, we had looked into optimization. What is optimization? What are the key components of optimization? How do we define a function and then optimize the function? Optimization is basically maximization and minimization of some quantity, as a result of which we get the best solution to that problem. And we also worked out some examples manually, and we also looked into how we can do this in our code. So today I will be moving on to optimizing, how to do optimization, how do we optimize parameters from various modules in BioCro. And along before I get into that, I will just refresh you through the basics of running BioCro as a summary to how we had summary to the BioCro, working with BioCro. So BioCro is a crop model that is mechanistic, which means that it makes use of physically based equations, and generic, which means it can be used for any type of terrestrial crops. And it is also a modular crop growth model. So history again goes like this, where initially it it started off as a software named Vimomap, and then it was updated by various researchers and well-known people to the 5 Pro version that we are using now. Mm, Miscanthus, willow, sugarcane, soybean, plants like these have already been parameterized in BioCro, and you can run the growth, find out the growth, and explore different processes of the plant using the soybean model and the other bioenergy crop models. And like I said, it is a modular crop growth model, which means that the various processes like photosynthesis, tomatal conductance, transpiration, carbon partitioning, soil evaporation, soil moisture stress, these all are represented as individual modules in BioPro, due to which if we want to explore a particular physiological process using the model, we can do that, like how we have been doing. So we have been focusing mostly on the FPCB model. We have gone through the Ballberry model. We have also gone through the C3 assimilation module, which is taking into consideration all of it is a coupled FPCB and Ballberry model. So this way you can either run the FPCB module around, or you can couple it with two models and run the two modules together. So this is what it means when you say that it is a modular crop growth model. And when it is simulating for the entire plant, it is taking into consideration the different microclimatic conditions within a canopy, so that it segregates the entire canopy in such a way, depending on the microclimate, like how much light is absorbed by each layer. And based on that, it simulates the various processes that are happening and scale it from leaf scale to the canopy scale. And further, it can also be scaled into a larger scale, uh, which is the area field scale. So just like how the paper, the BioCro describes its modularity is similar to how we can choose parts for a vehicle and send a specification to a factory and manufacture and send it to manufacture the vehicle. Similarly, we can also choose modules, give the necessary inputs, run it using BioPro and get the growth or biomass or total canopy, photosynthesis, canopy, transpiration, etc. by running this module. So when it comes to running BioPro, uh, we started with installing BioPro, which I guess is familiar to all of you by now. So, in order to run BioCro, we need to first load the package. So, BioCro is basically is a package that is available with in R. So, we just need to install that package and load the package BioCro by using this command library BioCro. Since we have it already installed in our systems, we can just load this library. One of the first commands that we explored is get all modules. So get all modules, you can see here when you type in this get all modules itself, it is showing you, showing you what are the arguments required in this function. So get all modules are listing all the modules. So you have to first provide a library name and the local module name. And here we'll get the get all modules library name, which will give us all the modules that are available in BioCro. So from here we get to see that there are around um 92 modules available in BioCro. The next command that I showed was the module in the most important commands, that is module info. So if we need to know about a particular module, we can just type in this function and given this module name here. And when you run this line, you will get information regarding what kind of module is it, the name of the module, what are the inputs required for this module, what are the outputs that you'll get for the module, is it a direct module, fixed step size, etc. So this is the format in which you'll require to give the name of the module with the package name, colon, the name of the module within double quotes. Then the next command was to obtain the response curves. We had talked about the various response curve, photosynthetic response curves like ACI, AQ, and AT curves. So in order to get all these response curves easily, we make use of this module response curve function, and we type in the module that we want to use to run this response curve. So here, if I'm using FPCB model to give the response curves, we need to give in the arguments according to that. So to know what I mean, it is definitely difficult to keep in mind what are the arguments for each module. So we can easily do that by giving question mark and the function's name, and then you will get which module you require to, I mean the details of the module that you want to use. So module response curve requires the module name, fixed quantities, and varying quantities. So these are the three arguments that you need to provide. So when you're providing the arguments within the parenthesis bracket, you can type it, you can give a comma and enter, or you can do it in the same line. It doesn't make a difference. We usually follow this as a practice in order to keep the code organized, because if you keep typing in here like fixed quantities and whatever the quantities, comma, and varying quantities, it is going to go beyond the screen. So in order to make our lives easier, we type in enter and we give the quantities over here. So the fixed quantities that is required for FPCB module, we can get by doing a module info for this BioPro FPCB. Again, This is not something that you require to keep inside your head because you can easily get it through this. So we'll copy all of these and here it needs all these parameters. Now we are going to have the ACI curve. So we will give the fixed quantities as a list. It is necessary that we provide this as a list. So inside the list, we'll give G star values like 100, comma, J is equal to 195, comma, KC, the Michaelis-Menten constant, say, let's give it 200, comma, K naught, let's give it 100, comma, OI is equal to the oxygen concentration to 10, RD is equal to say 3, and the TPU is equal to 10, and the VC max is equal to again 200, and alpha TPU is equal to 0, and electrons per carboxylation is a constant value of 4.5, and electrons per oxygenation is going to be 5.25. So all these except for these two values, electrons per carboxylation, electrons per oxygenation, and alpha TPU rate, all the other values can vary depending on the leaves, depending on the growth stage, depending on the type of plant. All are variable quantities. Now, once we have given all the fixed quantities, the next step would be to give the varying quantities. So varying quantities, we can again uh, define it as a data frame of consisting of the variable CI, which is intercellular carbon dioxide concentration, and we want to give values from 50, 100, 150, 200. So instead of doing this, we can also do like this, or instead of doing this, we can do an easier thing, which is use the sequence command and type in the number that you want to start in, comma, type in the number that you want to end with, the intercellular carbon dioxide concentration that you want the end value of it. And in the third index or for the third position, you type in what is the interval that you require. So I require an interval of 100, and then we close the bracket. So here the bracket is closed. Now we'll run this module. Say we give this result, and there is an error. If you see, after VC max, we haven't put a comma. So this is coming as an error over here. So we'll again correct it and then run it, and we get the result, which if you type in if C, you can see that for constant inputs of G star, J, KC, K naught, OI, RD, TPU, VC max, alpha TPU, you get outputs of Rubisco carboxylation, AJ, which is RUBP regeneration limited, net photosynthesis, TPU limited photosynthesis, and the these VC is the term usually. I think it's tau star minus tau star by CI or something like that, because it is done inside the model to make the model estimations easier. So these are the values that is obtained. I think AP value is coming a bit weird, so let's just uh, put the change the values and see. Mm, it's 200. Yeah, yeah, that's okay. Okay, so this is how we are getting the this is the result we got. And if you want to result, you can just click in here in the environment and you'll find the result for the vary. If you want to view this as if you want to view this in the form of a graph, we use this package called Lattice, run it, and then plot the graph like with the command xy plot the y-axis value that you want. So you want the y-axis value from this result, and the in the y-axis say you want a c value, and you also want the aj value. So you can just select it from here after giving the dollar sign, and then in result you need the TPU value, AP value, and in order to put the y-axis you give this tilde sign, and then uh, give the y-axis values that you want to select, which is the CI value. And then inside that we'll put type is equal to C P comma L, which means that you want the line to have points as well as it should be a line. And we give auto key is equal to true, which means the uh, legend will be shown. So when we run this, we get a graph, something of this sort, that is giving us the Rubisco carboxylation, the RUBP regeneration carboxylation, and the TPU rate. Now, if we want to plot the AN value along with this, you can also put the result result AN along with this, and then again run this, and you will get the AN line, which is going to be the minimum of let me see, N is blue color, but it doesn't look like it is here. Let's put result this value at the end. Probably it is being um, superimposed by the other lines. Yeah. So AN is this dark orange line, which is giving us that this curve is representing of the net photosynthesis, the minimum values of all the three limited phase uh, rates. Rubik's business carboxylation rate, RUBP regeneration rate, and TPU rate. So this is how you do an ACI curve. And in order to do an AQ curve, you need to do a module response with the module response with this module BioCro C3 assimilation. Since we are dealing with C3 assimilation, again, I'm not very sure about the what do I say? The spelling of this thing. So instead, so I go into this get all modules result, and I select out that module that I want, module of interest here, and then I paste it here, and just like the other modules, it needs the fixed quantities. So for fixed quantities, I'm going to use the soybean parameters. Oops. Soybean parameters, which has all of the values already. And for the varying quantities, I'm going to give a data frame of QABs, which is the x-axis basically. Again, a sequence from say 50 to 2200, again by an interval of 100. Now this module might show this line might show some error. Let's see that. Let's rectify the error. So you can see here it is throwing an error saying that stomata water stress is an input. tea leaf is an input, GBW is an input, RH is an input, temperature is an input. So the soybean parameters doesn't have all these values as input. So what we do is, along with this soybean parameters, we create a list of inputs which are required in extra, which is stomata water stress, which is going to be 1, since we are simulating it under not a stressful condition, 0 water stress. Tea leaf, we are going to simulate it at say 35 degrees Celsius. Then GBW, say it is around 2. GBW is around 2. Then there is RH, which is say at around 75%. So we get 0.7. RH is relative humidity, and temperature is equal to say 39 degrees Celsius. So this is giving us the list of fixed quantities. Varying quantities are this. Now let's hopefully, okay, so there are no errors. And okay, all these parameters are also required. So we need to put those two. Hmm. Okay, that is because in the soybean parameters, it already consists of CATM, GS minimum, G star EA, G star C, all of these. So we have to remove this. Okay, let's not use soybean then. Let's put all these parameters. Okay, this is going to be a lot of work. Okay, let's just do it. CATM is equal to 420. Then GS minimum is equal to say 0.001. Then G star EA is equal to, let me just put all the parameters here, list it all here. So it's here Jmax EA, Jmax C. So if you notice this Jmax EA, Jmax C are the temperature response constants or parameters of uh, that is coming in all the limited photosynthetic rate. Kc is the Michaelis-Menten's constant for carbon dioxide, and the temperature response parameters relating to it. Again, K naught is oxygen, Michaelis-Menten constant for oxygen, and the temperature response parameters corresponding to it is what we are giving here. We are not giving the direct value of that. So say let's take this as some value. 38792. G star C is equal to 39. J max C is equal to again 78906. J max C is equal to let's say 79. Same here, 56. KCC is equal to 57. To 69, and hopefully we won't require any other. I think we still require some parameters, but let's just run it and see. Okay, I haven't put a comma between anything, so again we are getting an error. It still needs more parameters. Just a second. There must be an easier way. So if you have any doubt regarding this, you can go to the BioCro GitHub repository. Their document is there. You can go to the practical guide to BioPro, and they have a lot of contents on that, on how you can um, you know put these. Yeah. So here with this trick, I think this thing will be solved. Within soybean parameters, yeah, there is something called a function called within. So within soybean parameters, your all the parameters will stay as it is, except you need to change these values which you want different. So here within the soybean parameters, we want all, if these are all the values that we want different. Actually, we didn't have to put in all of it. We can just give it like this. Okay, okay. Hopefully it will run this time. Um, okay, when we use this within, we don't need the comma. So let's remove the comma. Fine. So we got the AQ curve. So here what the mistake I was doing is I wasn't using this within command. So if you're using this within command and using the soybean parameters, then if we want to change some parameters within soybean, then we can put it within curly brackets, and the remaining parameters within soybean will be used by default. So soybean parameters are all of these. You can see here soybean parameters. When I give a variable of soybean uh, parameters, you will see that it is a list of 136 parameters with soil air entry. All of these are all the parameters available in soybean parameters. So in this, you can see certain parameters already here: G star C, G star EA, J max C, J max EA, KCC, KCEA, K naught C, K naught EA. All of these parameters are already there within soybean parameters. So we can just to run the model, we can directly use that and say we want to change the atmospheric carbon dioxide concentration, because now it is around 420. We will use 420 value instead of the 372, and say we are getting a B0, B1 value and all different, then we'll use that instead of the default value. So in order to just change the value, you need to use this within command, and within soybean parameters, say these are the parameters, variables that we want to change. Now, similar to how we plotted an ACI curve, we can plot an AQ curve also, where the data frame is AQ, and uh, we don't want all this because this is belonging to the ACI curve. And we can say these are the outputs, assimilation, type C, all of these, and the x-axis we want the QABS value. Now you will see. Oh, we have a weird AQ curve, and that too in the negative value. Uh, let's say I remove all of these. just put temperature. Okay, I'm not changing anything. Let me run it this day. Let me remove these ones. Okay, and then plot and see. Yeah. So some values that we entered are not right. That is why we got a weird curve. Now we have got a good looking curve of AQ, where you see this part is describing the maximum quantum yield, this convexivity theta, and here we know that this area until this portion, this area is the light limited area, and after this the maximum is obtained. So this will be giving the A sat value, and you will see the light saturation light saturation point is this point. So this is how you'll do an AQ curve. Now that was using module response curve. Now suppose you don't want a curve, but you just want to evaluate a module. Then we use this evaluate module command, and you can see over here itself, evaluate module, you need the module name and the input quantities. So again, evaluate module, we will run the, say we are running the Wahlberry module. And again, I don't know what is the correct name of the module. So I'll again go do biocro, get the list of all modules. And so let's take a simple function, biocro stomata water stress. So I'll put this here. And the input quantities required for stomata water stress is again, I have to check. So I'll do module info here and then use the same module's name to get what all are the input required. So here the input is soil field capacity, soil wilting point, and soil water content. So I'll put those inputs over here as a list probably. Input underscore quantities, because that is the name of the list of soil field capacity equal to say 0.5, comma soil wilting point is equal to 0.25, comma soil water content is say around 0.4. So what is going to be the stomata water stress is what we'll see now. We get the stomata water stress is 0.6, which is 60% water stressed. So this is how you use this function in BioPro, evaluate module, to just get the answer. Now evaluate module can be done for all these modules. So instead of module response curve, it has the capability of having a varying component. You just you cannot use that in evaluate module. In evaluate module, you just give the input, you get the output. But in module response curve, you can change the values of a particular variable and see how the other what is the final photosynthetic rate corresponding to the different values of the variable. So using the C3 assimilation module, we can also plot the ACI curve, we can plot the AQ curve, we can plot the AT curve, because this module response curve is having the capability of varying parameters. Now using FECB module, we can only plot the ACI curves because it doesn't take the input of light. So because we don't have an input of light, we cannot uh, use the value of generate an AQ curve using FECB module. And further, it is also not taking temperature. So without temperature, you can also not produce a temperature response curve. So this module is the best for producing ACI, AQ, and AT curves. Now, within the ACI curve, if you want to check if which is the carboxylation limited region, or RUBP limited region and TPU limited region, then you can use this module to represent that. Okay, so that was evaluate module. Now, another function which I didn't talk about in my earlier classes was something called get uh, get all quantities. Now, get all quantities, similar to get all modules, is going to give us all the quantities or all the inputs and output variables that is present in BioCro. So, if you do this, all quantities, get all quantities, again, get all quantities just requires a library name. You will see that all the quantities that are available in all of the modules in BioCro can be seen here. So you can see what is the name, the quantity name along with the quantity type and the module in which it is present. So ABA decay constant is an input in this ABA decay. If you see net assimilation rate is an input in Bolbury. C atmosphere is an input in Bolbury. Further again, C atmosphere is not just an input in Bolbury alone, it is also an input in C3 assimilation. So all these parameters corresponding to the modules and whether it is an input or an output in a module can be viewed through this get all quantities command. Now suppose you want a particular uh, quantity, you just want to know about where is the C atmosphere, which all modules are C atmosphere used, then you can C atmosphere all quantities, close the bracket, all quantities, dollar, the quantity name is equal to. So this is a condition that you're saying that all the quantities having the quantity name CATM be shown. Error in design. What's wrong? Okay, there should be a comma after this. So here, when you get that the concentration of atmospheric carbon dioxide is used as an input in Bolbury. It is used as an input in assimilation, canopy, C3 leaf photosynthesis, C4 assimilation. All of these modules, this input comes. Now, suppose you want the input for, say, let's say Q absolute, the Q absorbed, sorry, not absolute. Then again, you will use this thing and type in Q abs and run it. And you will see that Q abs is an input only in one module, that is C3 assimilation. So this is how you get the information regarding the quantities. Now, apart from that, there is also a website link that I've shared with you in the PDFs that I've sent, where you have all the quantities, what are the units, where it is used as an input, where it is used as an output. So you can either use that this while you're coding, you can use this information, because when you have it as a data frame right next to you, it will be more information. So it will be more useful. So this is how you get information from BioCro regarding what all are the quantities available. So this is just a refresher of how to use various functions in BioCro that are mostly required. So you can see here in one place I'm using this list, the data type list, a data structure list. In one place I'm using a data frame called varying quantities. Now if you see here, uh, if I run this line alone, I run this line alone, you can see I got a varying quantity data frame over here, which is having one column and around 22 rows extending from 50 to 2250. We had given the last number as 2250, all divided by an interval of 100. So that is what, this is how a data frame will look. This is how a list will function. Again, if I just run this alone, I will get a list of input quantities, and the list will look like this. I mean, it won't be in the structure of a data frame. Instead, it is having sublist soil field capacity, soil wilting point, soil water content with all these values. Then again, someplace I have used this vector. Again, when I just run this part of the code alone, you get a vector here on the right side. It's a character vector with indexes 1 and 2. First index is P, second index is L. So this is another data structure. So we had all looked into the basics of R, where this is one data structure, the data frame is another data structure. The list is another data structure that is commonly used in BioCro also. So this is how you deal with BioCro. Now I'll be moving on to optimization. So in optimization, now what I'm going to do is I want to get the quantities of this module, this result. The BioCro FPCB module. So in order to get the, suppose say I don't have the value of VC max. So if I don't have the value of VC max, I want to, but I do have the value of the ACI curve. So suppose I measured an ACI curve and I have the data with me. Now from that ACI curve, I want to obtain the value of VC max corresponding to that plant from which I measured. So in such a case, what I will do is, say, suppose this is the data that I have collected. Now, let me just save this data as an ACI curve. So in order to save this, I will convert this into a data frame. So let me see the structure of result anyways. Structure, result, and it is a data frame. So what I'll do is, I will write this into a CSV file. So I'm going to write this data called result into a CSV file as result. Okay. Now you see that this is the CSV file that I have, the module name and all of these quantities. Now here you can also see that it has the value of AC, AJ, AN, AP. Now hypothetically say this is a measured data. We have all the quantity with us, and these are the AC, AN data, and we want to obtain only the VC max. So in order to optimize VC max, that is to get the value of VC max, derive the VC max value from the measured ACI data, which is stored inside this result. What we'll do is, first we will define the vector that we want to optimize. The variable that we want to optimize is VC max. So we create a vector C3 or VC max vector. Let's put C3 vector. And since it's a vector, I will be using the command C bracket, and I'll put some random value of VC max as the initial condition. So initially, I'm just putting some value of a C3 vector and defining a vector like this. So I'll run this this line. After that, what we're going to do is we are going to define the function. So just like how we had defined a function earlier in the last class of simple linear equations with a single variable or multiple variables, we'll define a function here called function to optimize VC max. Okay. So that function, in order to create a function, you should use this function command, is a function of the C3 vector. not only the C3 vector, but we have to run the module FECB. So for running the FECB module, you need fixed quantities. So it is a function of the vector, the fixed quantities and the variable quantities. So these are this function is going to depend on the C3 vector. It will also depend on the fixed quantities that we have. It is also going to depend on the variable quantities that we have. Now, inside this function, we have to define all the fixed quantities. The fixed quantities is equal to all the fixed values that we have used to run this module, the fixed quantities that we have used to run the FECB module. Now, if you see here, we will put this here. So these are the fixed quantities with which we ran. But then we don't have the value of VC max. We are going to find the value of VC max. So VC max, we will the value we will replace with this vector, which says that this definition, which says that VC max is the C3 vector that we have defined here, giving an initial value. And when we run this function, or when we maximize or minimize this function, we will get what is the value of C3 vector, which is the optimum value. So these are the fixed quantities. Now, next we need to give the variable quantities. So variable quantities is equal to the variable quantity that we are giving here is a data frame of CI values. So that itself will give us the variable quantity over here. A data frame of CI value. Okay. And we are going to use the module response curve with the FECB, which is going to be the module. The fixed quantities is going to be the fixed quantities that we have defined here, and varying quantities is going to be the variable quantities that we defined here. So varying quantities is equal to variable quantities that we defined here. So these are the three arguments required for module response curve. We are going to be generating the, first we will define within this function, we will take the values of fixed quantities. Second, we'll take the values of variable quantities. We will run the module response curve. And after running the module response curve, we will read the CSV file. Oh no, let's do. We need to read the observed data. Now the observed data is going to be the result. So the observed data is going to be the result dot. Now we want to get the values of VC max. So let's choose the observed assimilation rate, or the observed photosynthetic rate to be the carboxylation, maximum carboxylation rate. So observed data is equal to, we'll read the observed data, that is from result, we will get this observed data. Then the predicted, here we will define this module as predicted. Okay. Now the predicted data is equal to the predicted AC. Okay. Now FECB module is going to predict an AC using all of these input. Then observed data is equal to having the AC that we have already saved. Hypothetically, it is the measured data. And then our objective function, what is our objective function, is going to be the error between this observed data and the predicted data. So what we have to do is minimize the error between the observed value and the predicted value, such that if there is no error between the observed value and predicted value, then that corresponding VC max that is resulting in an AC value that is very close to the observed value will be the value of the parameter that we require to optimize. So we will find out the error between the observed data minus the predicted data, square of it, because error can be positive or negative. So usually we find the square of the data, and the final SSE, that is the sum of the square of the errors, will be sum of all the errors. Now, why we find the sum of the square of the errors is because this one quantity has to be a single vector value. That is single value it has to be. Here, when error is being calculated, error will be calculated for each value corresponding to AC, each value corresponding to predicted AC. So it will have a number of values. Now, what we want to optimize or what we want to minimize is the sum of all these errors. We'll get a number of errors over here. Then we'll find the sum of all the errors. And if that sum of all the errors is minimized, then we say that we have got the optimal parameter. And then we will return the SSE. So we are returning this line, that is all of these will be calculated within this function. When it is run, first the fixed quantities will be loaded, variable quantities will be learned. The model will predict it based on the fixed quantities and variable quantities. Then the observed data will be read. The predicted data will be extracted from this module. And then the error between the observed data and the predicted data to the square will be found out. Then sum of all of the square errors will be found out. And then if the sum of square errors is minimized, then if it is one value is found out, and if it is less than the previous iteration, then it will stop. Otherwise, if it is more than the previous iteration, then again it will go back to this place and find out the fixed quantities, variable quantities, and this loop will continue until the function, this one, is minimized. So our objective function here is the sum of square of minimization of the sum of square of the errors. So now we have defined the function. Now what we want to do is run the optimization. So in order to optimize VC max, we have to use the package library doptim. Now last class we had looked at optim. There is another package called doptim which you will have to install, and using that function we will run this optimization. So optimize VC max, we will use this the command in doptim. The command in doptim, if you can see here, requires the function that has the objective function, the lower limit, the upper limit, and some control limits. So here the function is going to be this function that we have defined, function to optimize VC max. Now the lower is going to be again the C3 vector we have defined over here as initial 100. So the lower limit, say let's keep it 20 or just 20 because it is just one value. Then upper limit is going to be say let's keep it 300. By default 200 iterations will run. So we will run this iteration. So first let's run this vector. Then let's run this function. We loaded the library. And when you start optimizing here, all this iteration is happening over here. You can see that 22 iterations happened. The sum of the square errors is this. First, it will give for the initial value of 100, the sum of square errors is 105. Then it takes a very good value, which will give only this much error. Then it will give only this much error. Then as you see the value of error is becoming very small. And finally, when 200 iterations are over, you will see in all of these iterations, the error is not changing because it is already got the optimized value. So whatever iterations are running is giving us the optimized values. So once the iterations keep running like this, So 200 iterations is the default. You will get, and here optimize VCmax is the variable that we have for the optimized parameters. Uh, when you click on this optimize VCmax, we have a list with two components. One is two elements. One is optim and one is member. So if you go to the optim and you see the best member, you will see that the parameter, optimized parameter value you're getting is 200 and 200 iterations. And then if you see, this is the data of all the information that we gave inside. The lower limit was 20, upper limit was 200. In each iteration, these are the values they have got. Now if you see here, see, in each iteration, so if in the beginning, the first iteration yielded this value of parameter, this one. So, and using this parameter, the FPCB module was run, and AC was produced, and that AC minus the observed AC was checked, but they found that the error is high. So they again took another value. They took this value, they took this value, they took this value, and they were seeing that error kept on increasing. So finally, they took this value until they reached this value, which was not giving any error. So if you see here, in the earlier result also, we had given the value VCmax is equal to 200. But here we gave the value, initial value 100. Through optimization, the optimizer found out the most appropriate value of VCmax over here is 200, which is again equal to the value that we got. Now, if you see here, you can plot how does the observed, sorry. Okay, that is going to give the same answer. Now, this is how we optimized one parameter. Now, we want to also say, suppose we want to optimize G star value, all the parameters that are corresponding to the carboxylation limited. So, we don't have G star value. So, we are going to use this as a vector, C3 vector. We're going to name this one. J is RUBP limited, so we are not doing anything with that. Then C3 vector 2, that is Kc. Again, K0 is also a carboxylation related parameter, so we want to optimize this value also. Then Vcmax is also another carboxylation related value. So we are going to optimize 4 values. So in that case, we have to define initial values for all of these, G star, Kc, K0 and Vcmax. So let's give an initial value of 100 for each of them. Yeah, we gave an initial value of 100 for each of them. Now, in the lower also, you have to give a lower for all of them. So let's give 20 for all of them and upper as 300 for all of them. Okay. Now again, run C3 vector. Run this function. There are no errors as of now. Now optimize this and sorry, I just forgot to put a bracket here. So now run the optimization. What happened? Okay, sorry, here comma is also not there. So when you see the optimizer is running a bit slowly because unlike the earlier time, we had only one parameter to optimize. Now it has got 4 parameters to optimize. So it is taking much more time than the first how the first optimizer ran. So this will take maybe like 2 or 3 minutes. So now itself you can see the best value of iteration. This is the error that is found. Now itself from the error that was starting at 469, reduced to 244. Okay, while this optimization runs, please give me 5 minutes. I'll So often when we are optimizing for in during field experiments, we take replicates of curves. Now, if you want to measure ACI curve, we don't just measure one curve. We'll take multiple replicates of ACI curve at different conditions, and then we have to optimize the parameters for each of these curves. So this you can see for just one curve, corresponding to one curve, running four parameters is taking more time than we had run it for one parameter. So often these optimization programs run for days and sometimes for weeks also before we get a final result. So it is important that we give all the quantities carefully because the once optimization start, we'll get the value only at the end. And if it is at the end that you're realizing, oh my God, this is a wrong value that I gave, then all 3-4 days will go waste. So you can see how the error is reducing from 0.001 to 0.0023. And you can see what are the values of the parameters corresponding to it. So by default, doptim has 200 iterations that will happen. Now, say if your error is not being reduced, if your error is still large, then you can increase the number of iterations to 400 or 500 or 1000 by giving this control. If you give something like the optim control and iter max, the maximum iteration is equal to 1000, then this iteration which is running 200 times will run 1000 times, and then it will give you a more finer result. So here if our error is 0.00232, say we want it to 0.000001, then we can run it for like 500 times to get the results. Now optimize VC max. If you see here, the best member gives us a value 100, 262, 82, and 200. Now, if you go back to the result that we had, we will see that for a value of 100, 300, 100, and 200. These were the values for which we simulated and we got the AC curve. But here they are saying the best value is 100, 262, 82, and 200. So these are another set of values. The G star and the VC max value is similar to what we had used as an input. But the KC value we gave us 300 and K naught value we gave us 100. But the optimizer is saying 262 is better and 82.4 is better. So let's see, let's generate a curve result 2 with this new parameters that we got and see if our AC values are varying. So we will run the same module, the same thing. with the new values of given by the optimizer. So if you see, I have, let's change the values over here. G star is given as 262.3. No, sorry, G star is 100 only. It is given as 100. KC is 262.3. K naught is 82.4. And VC max is again 200 only. They're giving 200.1. Let's use that value there. 200.1. Now let's run this and then plot XY plot for the first result that we had using the initial value of 300, 200, 100 and 195. No, 200 plus result 2 AC. In the y-axis, we'll have CI. Again, type is equal to point, comma, line, auto key is equal to. When you plot this line, you will see that both these curves are coinciding. So that means that for the value of KC, K naught as 200 and 100, and the value is changed to 262 and 82, finally the carboxylation limited rate is going to be the same. So now let's change either one value and see. Let's see if what happens to the result when we make this our initial when we make this 200. We'll plot this and we will see that, see, the curve varies. So result 2, instead of giving 262, we went back to the old value 200, but we didn't change the K naught value. So what happens? The curve is going at a higher rate. The carboxylation rates are being higher. So these are some inferences we can draw. We can say, I mean, if these two parameters are varying proportionally, it means that we still can get the same AC curve. So it doesn't mean that when two AC curves are the same, it doesn't mean that the parameter values are the same for each of them. Some can have parameters corresponding in this way, which is also giving the same AC rate. So this is an important inference you can draw from optimizing a curve. And just like how we optimize 4 parameters, you can optimize all of the parameters if you have the proper observed data with. So this is how we optimize in BioCro to obtain the parameters. Now, left. Okay, so if we need to write, now we got all of these results. So if we need to write this results into a file, we will go to write.csv. We want to write the result, result 2 also into the same folder, and you can write result2.csv. So you want to see these results even after you close the program. In that case, you can see that inside this BioCro codes, I have saved the result 2 and result 1. And when you open those, it's a lot. Let me just share this. So you can see that we get these results in the form of Excel files also, if you write it. So these are the curves. I think you weren't able to view it. You should have told me, but it's fine. So these are the curves when we change the 200 value. Now, these are the curves when we don't change the value to 200. And we use the original value 26.3. So you see, um, this is how the curve will be when you plot it. So that was about running optimization in BioCro. So I think that will wind up the BioCro portion. I would also like to run a summary, start with the summary of what we have gone in all these sessions. So BioCro, as of now, I have looked into BioCro. What are the functions that you can use in BioCro, how you can get the module response curve, how you can get all the quantities of a module, how you can get the informations of a module, how you can get quantities of all modules in BioCro, how you can get all the functions that is all the modules that are available in BioCro, how to create a function to optimize the parameters in BioCro, and how to use this doptim. Previous classes, we had also looked into running simple optimization problems. Also, we have worked with the data structures, introduction to working, I mean, basics with R, how to use what is data types, what are data structures, how can you create a vector, list, data frame, matrix, array, what are the differences between all of these data types, how to create a function. So all of these have been covered with respect to the coding portion of this course, plant modeling course. And as far as theory is concerned, we started with the introduction where we went a bit into what is plant ecophysiology. So, what are the different branches available in plant science? Out of which plant ecophysiology uh, is a is the science that deals with the plant physiology, combination of the plant physiology and plant ecology, and what is its importance when it comes to, I mean, why is it required by all of us to learn what plant ecophysiology is, as particularly as people in the water resources sector. Whenever we simulate hydrological processes, plant is an important component. So it is very important in terms of engineers dealing with any kind of works with hydrology. Then their plant physiology also deals with a lot of biochemical reactions, and so again, um, uh, again, plant and soil are very interlinked with each other. So how soil scientists can use this knowledge regarding plant ecophysiology when making various uh, studies regarding the transport of water in soil, the nutrient transport in soil, etc. Then we went a bit into the plant structure, not a lot, but because all of you are already biologists and people, I think from a good background of botany, we just brushed through what is the structure of a plant, what is the structure of a leaf, how is it when we go inside a leaf, and where exactly is the photosynthesis happening, which is basically the palisade cells that comprises of the chloroplast. And then again, inside a plant cell, there are mesophils, spongy mesophil cells through which air is, carbon dioxide, oxygen, and gas is circulating. In the lower epidermis, you can find the stoma through which gas exchange is happening. So all of these we looked into, we went through a bit in the leaf cross-section part. In the palisade cell consisting of chloroplast, we looked at the structure of the chloroplast, and we have these thylakoid membranes where the photosynthesis is happening. And the second module was majorly about C3 photosynthesis, C4 photosynthesis, and CAM photosynthesis. How C3 photosynthesis and C4 is different. C3 photosynthesis, the process of, okay, let's just go into C3 photosynthesis. So the photosynthesis in general is divided into two categories: light reaction, and Calvin-Benson cycle or the Calvin cycle. And the light reaction is occurring in the thylakoid membrane. Calvin cycle is occurring in the stroma part of the chloroplast. And light reaction converts the light energy into chemical energy represented by the ATP molecules and NADPH molecules. And the Calvin cycle happening in the stroma makes use of this ATP and NADPH to produce glucose molecules through a series of reactions. So in the light reaction is basically an electron transport chain where when electrons get excited in the presence of the photons from light, and this excited electrons that is formed as a result of splitting of water molecule in the oxidative center of the photosystem 2 um, follows an electron transport chain from PS2 to PS4 to system 1, where one reaction happening is the reduction of NADP to NADPH, and another phenomenon happening is the concentration gradient setup as a result of hydrogen ions concentrating in the lumen portion, which drives the ATP synthase out into the stroma again, during which ADP is reduced to form the ATP molecules. Then these molecules are these are the energy molecules that is utilized in the Calvin-Benson cycle. In the Calvin-Benson cycle, three important phases occur: carboxylation, RUBP regeneration, and TPU utilization. So I think a majority of the portions in this plan modeling course has been involved, has majorly focused on these three processes: carboxylation, RUBP regeneration, and the TPU limited photosynthesis. Then during the Calvin-Benson cycle, again, this molecule called RUBP, ribulose by 1,5-biphosphate, is combining with carbon dioxide with the help of this very important enzyme Rubisco, which is resulting in the formation of PGA molecules. And these PGA molecules are again reduced into GA3P. GA3P is what is called the triose phosphate. This triose phosphate is then when in the cycle, the triose phosphate molecules help in the generation of the glucose molecule, and some of it, the remaining of it, goes back in regenerating RUBP. So the electron transport chain is what is resulting. The light dependent reactions are what is resulting in providing energy to the Calvin cycle, and this Calvin cycle is making use of this energies, that is ATP and NADPH, and converting it back to ADP and NADP plus and the hydrogen ions, which again take part in the light dependent reaction to keep on generating energy for this entire process to happen. So and in comparison to this Calvin cycle in C3 photosynthesis, we see that the enzyme Rubisco has a high affinity towards oxygen also, apart from carbon dioxide. So at times what happens under certain conditions of when the oxygen concentration is really high in the in the atmosphere, or when the temperature is really high, the Rubisco the RUBP, instead of reacting with carbon dioxide, will go and react with oxygen. And as a result of which, PGA is formed, but another molecule called the phosphoglycolate is formed, which cannot take part in this Calvin cycle. So what it does is, it goes through another different set of reactions, not only happening in the chloroplast, but also in the peroxisome and mitochondria, to convert this phosphoglycolate into PGA so that it can again be a part of the cycle. But unfortunately, during this series of reaction that is happening in the chloroplast, in the peroxisome and the mitochondria, carbon dioxide is utilized for forming the intermediate molecules or the compounds or the whatever substances that is formed during the set of reaction. This is resulting in the wastage of the carbon dioxide molecules, which otherwise could be used to fix carbohydrates, I mean, to generate carbohydrates. So even after this phosphoglycolate is getting converted to PGA, all of this PGA is converted to G3P, which is not sufficient to generate glucose molecule, and neither is it sufficient to regenerate RUBP. So in altogether, as with respect to photosynthesis, this process of photorespiration is considered a waste process because it is wasting carbon dioxide, it is not generating glucose molecule, it is not regenerating RUBP properly, but it is also making use of all the energy that is utilized during light dependent reaction. So in order to make it more efficient, there was an evolution of photosynthesis, which is a C4 photosynthesis. Then we talked about the process of dark respiration, where there are three major processes happening. Uh, as a part of this dark respiration, which is also the respiration that is occurring. This is called dark respiration because this doesn't need light to occur. And this is a very important kind of respiration. Unlike photorespiration, this respiration process is what is helping in the growth and maintenance of plant cells. So it is a very important kind of respiration. Oxygen is released during this respiration. Carbon dioxide is released during this respiration process. So it occurs in three series where this process called glycolysis happening in the cytoplasm, where glucose is broken. Then there is this Krebs cycle that is occurring in the mitochondria and converted into the Krebs cycle converts this into releases carbon dioxide and produces some other energy products. And in the electron transport chain also is occurring, which is helping in pumping the protons across the membrane to create a proton gradient, just similar to the electron transport chain in the light dependent reaction. So this is a very important kind of respiration, unlike the photorespiration, which is providing around 38 molecules of energy molecules for the maintenance of the plant as well. So this is the Krebs cycle and the electron transport chain, glycolysis, and the Krebs cycle here that I just now explained. So you can see here how it is, what is the net quantity of um molecules that are produced over here. I'm sorry, the energy that is produced in the end. Okay. Okay. And a Krebs cycle is also called the citric acid cycle. Then we looked at what are the limiting factors when it comes through C3 photosynthesis. The Rubisco can be a limiting factor because during the carboxylation limited rate, the activity of the Rubisco, if it is less, then carboxylation, that is the fixation of carbon dioxide will reduce, which is again in turn going to affect the entire net photosynthesis. Similarly, RUBP can be a limiting factor. That is, the regeneration of RUBP can be a limiting factor. And the utilization of triose phosphate can also be a limiting factor. So, triose phosphate works on a give and take policy where when they uh, give a triose phosphate molecule for the manufacture of glucose, the cytoplasm has to push back a phosphate molecule, which will take part in the light independent reaction. So if triose phosphate is not utilized properly, then this phosphate molecule again also will not be provided adequately, which is again going to limit that reaction. So these three processes, maybe the Calvin cycle is a very important cycle based on which all of these equations and models have been developed. So a deep understanding of this Again, this is also just brushing through the superficial part of it. If you want to go into each of these limited phase, there are a lot of information or study that has to be conducted for each of these processes, particularly the RUBP regeneration limit, which is, I feel, sometimes not very clearly represented in the FECB module. It is clearly represented, but again, it's a bit confusing for me to related, because it is not just happening in, I mean, ACI curves are not, the ACI curves are not, are dependent on this RUBP regeneration limit, and some factors are also dependent on the AQ curves. So it is a very confusing part for me. So I think that is one part that requires to be studied a little more. Then we went into one of the most important parts of this chapter, which is the photosynthetic response curves. So photosynthetic response curves, three types are there. One is the carbon dioxide response curve. So carbon dioxide response curve is nothing but the response of photosynthesis to changing carbon dioxide concentration in the atmosphere. Now, atmospheric carbon dioxide concentration is something that governs, or how much is the concentration of the what will be the concentration of carbon dioxide with inside the leaf. So the plot of the net photosynthetic rate to CI values is what is called the ACI curve, or carbon dioxide response curve. And as you see here, the three limitations that we studied, Rubisco carboxylation limited phase, RUBP regeneration limited phase, and triose phosphate utilization limited phase. All of these are represented in the ACI curve. So the first part, where the curve is increasing with increase, the photosynthesis is increasing with increase in CI value, represents the Rubisco limited carbon dioxide phase. So any value of of photosynthesis that is happening at lower values of intercellular leaf concentration will be depicting the carboxylation limited rate. Now, there is a point in the curve where this curve is transitioning from a sudden, a steady increase in photosynthetic rate with changing CI value to a point where it is not increasing that much with CI value. So the increase drastically reduces, the slope drastically reduces. So this space where there is a transition from this steady increase to no increase is called as the RUBP regeneration limited phase. So here what you see is Rubisco is the governing factor over here. So the activity of Rubisco is what will define the photosynthetic rate in this region. But when you see the curve has got a part like this, it means that RUBP is the limiting factor here. Whether Rubisco has high activity or low activity, for the values of photosynthetic rate corresponding to these values of CI will change based on the regeneration of RUBP. So that is what it means when something is a limiting factor. That is the controlling factor over there. So this part of the curve, or these values of photosynthetic rate is governed, or is affected more by the RUBP regeneration part of the Kelvin-Benson cycle. Then again, when you find that the curve is going flat after a particular value of intercellular carbon dioxide concentration, we see that it is because of the triose phosphate utilization rate. Here again, whether the leaf has got high activity of Rubisco, or whether it has got high affinity for carbon dioxide, or whether RUBP regeneration is happening sufficiently, it doesn't matter. Here, if the triose phosphate, this portions of the curve is telling us that the triose phosphate is what is governing that rate of photosynthesis at high rates of intercellular carbon dioxide. The triose phosphate utilization, whether it increases or whether it decreases, is what governs the photosynthetic rate, or controls the photosynthetic rate that you see over here. Okay. Then we moved on to the light response curves. So in light response curves, there are again similar to inferences drawn from the ACI curve. the plots of net carbon dioxide uptake with respect to the amount of photons that are absorbed by the leaf is this plot of AQ curve. A for net photosynthesis or net carbon dioxide uptake to PPFD, that plant available radiation, photosynthetic photon flux density. All of these can mean the term in the x-axis. So the leaf might be receiving some amount of light. We had seen that all of that light is not used by plants or absorbed by plant. Mostly it is the short wave, the light in the short wave wavelength that is absorbed by plants, and not all of it again. Only around 45%, 50, 45 to 80% of the light is absorbed. So the number of photons is calculated. The moles of photons is usually calculated to quantify the amount of light absorbed by the leaf or a plant. So that is what is shown in the x-axis. Now from an AQ curve again, similar to an ACI curve, we can get the rate of respiration again, which is giving us indication of the fact that this point where the net carbon dioxide is equal to 0 gives us when um, yeah, when will the net uptake of carbon dioxide is equal to 0, indicating at what light will neither photosynthesis nor respiration will occur. So that point is called the light compensation point. And again, in the light compensation, in the AQ curve, we see that there is an increasing rate of photosynthesis with increase in plant available radiation, and it will linearly increase for some time. You can see this portion of the curve has a linear increase. Then after that, it will slowly start declining. That is, the increase will stop slowing down a bit until it reaches a point where beyond which there is no increase or decrease. Now, this part of the curve that you see here is what is defined by the convexivity term in the value of J, which again I'll come back to. And this part of the curve will give us the maximum quantum yield. That is, if you see this a curve that is having a higher slope compared to a curve with a lower slope, the amount of net carbon dioxide uptake happening is more, whereas here it is less. So basically, it is the light use efficiency of a leaf or a plant. The information regarding that you will get from the sloping portion. And this convexity factor is a factor that is governed by the resistance to carboxylation as well as resistance offered by the boundary layer, which is given by the non-rectangular hyperbolic equation. Again, I don't think I have the time to go into the explanation of the non-rectangular hyperbolic equation. So there will be a follow-up course in this, and probably I'll explain in that. So this part of the curve, the convexivity factor, is governed by the resistances offered to the carboxylation and to the boundary layer, resistance offered to carboxylation and resistance offered by the boundary layer. Now, after this maximum photosynthetic rate, you will see that there is no increase with light. This is indicating that beyond this photosynthetic rate, whether you give more light or less light, whether you give any, not less light, if you keep giving more and more light, it is not going to be of any use to the plant. Photosynthesis, in terms of photosynthesis, no photosynthesis will occur. So this part of the curve is actually giving us information. So if you analyze these values of photosynthetic rate, then you can get that this part of the curve is either governed by the Rubisco, RUBP or TPU. It is not governed by light. So this is the portion of the curve which is not affected by light. So in all of these values, when all of these values of net carbon dioxide uptake is considered, light is an important factor that is controlling the rate of photosynthesis. But beyond this point, light, whether you give how much ever amount of light to give, it is not going to impact the rate of photosynthesis. So this is the important inferences that you can draw from an AQ curve. Then we looked into AT curve, which is nothing but the response of photosynthetic rate with changing temperature. Now, this is an important curve when it comes to a lot of parameters in photosynthesis, which is a function of the leaf's temperature. So, if from this curve, we can derive or we can obtain a lot of uh, parameters that are helping us obtain values which are used in all of these equations, that is corresponding to the leaf temperature. And you can see this pattern that is generally followed by when it comes to a temperature response curve is that we will keep increasing with increasing temperature, but at a certain point it will reach the maximum value, and after that value of temperature the curve is going to reduce, indicating that it is not very safe to provide high temperatures to plant. I mean, if there's a general notion that at high light intensity, if the temperature is very high, then the plants will photosynthesize more. But that is not what actually happens at a very high a very high temperature condition. The activation, the rubiscose activity rubiscose activity will decline because of this, and the photosynthetic rate will decrease. So when it comes to a climate analysis or climate change impact studies, this is a very important inference that we can draw, saying that in the future, as a result, the temperature is going to be really high. So under such conditions, how will plants respond to this high increasing temperature is a very key study that needs to be conducted to see how this temperature is going to impact plants in the future scenario. So this is what an AT curve, what we can infer from an AT curve basically. Then we moved on to this very important equation, Farquhar-von Cameras-Berry model. In this model, the equations that are depicting carboxylation rate, RUBP regeneration rate, and the triose phosphate utilization is explained, and it is given by this first equation, where the minimum of all of these three values, that is three limited phase photosynthesis, is considered and subtracted by the growth respiration or dark respiration, which gives you the net assimilation rate. And Rubisco limited photosynthesis is calculated using this formula. Vc max is the maximum carboxylation rate of Rubisco. Ci is the intercellular carbon dioxide concentration. Tau star the compensation point, which is basically the point at which the net carbon dioxide uptake will be 0. The carbon dioxide concentration at which the net carbon dioxide uptake will be 0. And Kc is the Michaelis-Menten constant for carbon dioxide. K0 is the Michaelis-Menten constant for oxygen. So that constants are giving us an idea about the concentration of carbon dioxide and oxygen. So, and this is how the Rubisco limited photosynthetic rate is simulated. We are familiar with this value of this AC from Biochrome model, where the module FPCB is predicting these values. Then we have the RUBP regeneration limited photosynthesis, which is given by this equation J into Ci minus tau star by VCCi plus 2 E naught into tau star. Here J is the rate of electron transport, Ci carbon dioxide concentration inside the leaf, carbon dioxide compensation point, Ec is the electrons that are used for carboxylation, and E naught is the electrons that are used for oxygenation. Now Ec and E naught are in their values are used as 4.5, and another set of value E naught is used as 5.25. That is because for every carboxylation corresponding to that, the equivalent amount of that is photorespiration or oxygenation that has to occur is 2 times. So for every carbon dioxide molecule, 2 types of oxygenation has to it takes 2 times of oxygenations. Sorry, every carbox the energy used for carboxylation of one molecule of carbon dioxide is corresponding to the energy used by 2 times of oxygenation. Something that I think I'm messing it up, but that is how the relation is. And then this value of J gives the rate of electron transport, but J itself is a parameter that is impacted by light, the convexivity factor, the maximum electron transport rate, and also the quantum yield. So the 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 equation over here that is depicting the intensity of light that is absorbed is again a function of the maximum quantum yield, then the fraction of absorbed light reaching photosystem 2, and the light reflectance or absorptance. And here theta is again this convexivity factor. So using this equation, you can find out J value, and all of these value are again temperature dependent. If you see, Jmax is temperature dependent, theta psi is temperature dependent, phi psi is all temperature dependent. So if we have a temperature response curve, then we can find what is the value of theta corresponding to different temperature, what is the value of phi that is corresponding to different temperature, what is the value of Jmax that is corresponding to different temperature. So all of them are temperature related parameters. So temperature is a very important parameter when it comes to all of these equations. Same way, Vc max is also temperature dependent, Kc, K naught, tau star, all are temperature dependent. This you can see in that C3 assimilation module where we are giving a lot of constant Kc, C, Kcea, then again K naught C, K naught Ea. These are all the temperature response parameters obtained from that Arrhenius equations and all, which are temperature response functions. Then there is the TPU limited photosynthesis, where TPU is photosynthesis is directly proportional to the maximum TPU rate, and the minimum of the three will be considered for estimating the net photosynthesis. So in the next class, I'll be summarizing those portions after this part where we're going into the stomatal conductance, the leaf energy balance, gas exchange, and all of that. So again, next class is just going to be a summary class. So if you have any doubts during the session, please feel free to ask and make use of the last class. Again, today's I'll end it here. If you have any doubts, please feel free to ask. Otherwise, you can leave the session. Thank you for attending today's session. See you on Saturday. Thank you. Night. Good night. Thank you, Maya.